astounding uses of genetic technology. Hello and welcome back to another video here on UpTech channel. Genetic technology is changing the world as we know it. As you watch this video, scientists are working on fascinating ways to modify DNA. Recently, a form of advanced gene editing technology known as CRISPR has opened up new avenues of genetic experimentations. CRISPR is held in such high regard that in 2020, its creator were awarded the Nobel Prize in Chemistry. In this video, we will take a look at some of the most important technology advances in genetic engineering. Stay tuned! Modified goats produce cancer drugs in milk. Scientists in New Zealand have created genetically modified goats that produce cancer drugs in their milk. The goats have been specifically altered to create cetuximab medications, which is used to treat cancer in the colon and lungs. Currently, the drug can cost as much as $13,000 a month without insurance. Scientists hope their new method of production will help lower the price, thereby making it more accessible. Manufacturing cetuximab is an expensive process. Its elaborate chemical structure means producers have to rely on proteins from inside mice cells to cultivate the drugs. But these genetically modified goats have offered the pharmaceuticals industry a way to mass-produce cetuximab. It's a lot more economic to make cetuximab in animals because their mammary glands can produce large amounts of proteins, explained by Goss Label, the researcher in charge of the project at New Zealand's Ag Research Institute. Scientists stole data inside living DNA. Data storage is difficult. There is not enough space to store data, but that's nothing compared to what scientists are working on currently. Scientists in New York have come up with a new method that uses gene editing to store data in the DNA of live bacteria. In 2021, researchers at Columbia University demonstrated that live E. coli cells can store up to 72 bits of data. And its core, a data file consists of a long line of ones and zeros. The scientists were able to encode ones and zeros into the E. coli DNA by inserting specific genes into the cell. They were even able to write the simple message "Hello World" to the DNA of an E. coli cell, then decode it in by sequencing the DNA. DNA is surprisingly well suited to data storage. Biological proteins can store a massive wealth of information. Scientists estimate that if a strand of DNA were the size of a grain of salt, it could hold the equivalent of 10 feature-length movies. What's more, the technology needed to read and write DNA is becoming increasingly powerful with time. That said, DNA storage is still in its infancy, which means it's unlikely to take off anytime soon, increasing the lifespans of dying mice. Scientists at Harvard University have extended the lifespan of dying mice using gene editing, increasing their life expectancy by more than double. As part of the study led by Professor David Liu, mice were given progeria, a rare disease that causes aging in children. On average, children with progeria live to the age of 14. The condition is caused by rare genetic mutations and cannot be treated using regular gene therapy. Instead, the Harvard team is developing a way to change the fundamental coding of the progeria sufferer's DNA. This technique was trialed on the terminally ill mice, and it significantly improved the length of their life. The rodents, which were expected to survive 215 days, were on to live for a median average of 510 days. Liu and his team hope to use these findings to develop an effective treatment for progeria and similar genetic conditions. Gene therapy enhancing vision. Scientists have recently discovered a form of gene therapy for sight loss that, when injected into one's eye, improve visions in both. The genes travels from the injected eyes into the untreated eyes. Although eye specialists are unsure about the implications of this discovery, the scientists were attempting to treat a condition known as Leber's hereditary optic neuropathy, or LHON, a form of progressive sight loss mostly found in young men. This rare type of blindness stems from a genetic mutation that attacks and destroys cells in the eye's retina. As part of a recent trial, 37 patients with LHON received gene therapy injections into one of their eyes. But remarkably, after 2 years, 29 of the patients reported the improvement in visions in both eyes. At first, the scientists were taken aback by these results until they discovered that the gene therapy viruses were sneaking out of one eye and into the other. Repeating the experiment on macaque monkeys, they found the genes were traveling down the optic nerve of one eye, crossing over to the other optic eyes, then traveling into the other eye. Harmless but with no horns. Researchers have found a way to create hornless bulls by editing the DNA of the father. This new method provides farmers with a painless alternative to current dehorning techniques. Currently, cattle need to have their horns physically removed. This is a lengthy and difficult process that can be extremely painful for the bull, but it does need to be done. Not only are the hornless bulls less likely to harm other animals, but they are also easier to transport and take up less room at a feeding trough. In 2016, two baby bulls were born with the genetic mutations that means they would never grow horns. This was achieved by introducing a short strings of DNA into father cells. After analyzing DNA from three bulls, scientists confirmed that the genetic alterations had been passed down to the young cattle without causing any accidental side effects. We have demonstrated that healthy hornless calves with only the intended edit can be produced, and we provided data to help inform the process for evaluating genome-edited animals, explained Alison van Ininem, an expert in animal science working at the University of California, Davis. Cows made more resilient to heat stress. With rising global temperatures, cows begin to feel the strain. Bovines are particularly susceptible to heat stress. If left in the blazing sun for too long, cows start to lose their appetite, produce less milk, and are less likely to conceive. As you can imagine, the knock-on effects for farmers can be terrible. Heat stress is said to cost the US dairy industry $900 million. In poorer countries where farmers may only own a few livestock, it can be the ruin of many. But now scientists in New Zealand have found a potential solution to this cattle-based conundrum. They are using gene editing techniques to change the color of the cow's coat. By altering their pigmentation genes, the researchers managed to lighten the dark, heat-absorbent hair of common dairy cows. Holstein Friesian cattle are usually white with jet black patches, but the genetically altered cows were born covered in light silver-colored markings. The researchers hope to refine their research using DNA from tropical cattle that are more resilient to high temperatures. Overweight mice lose body fat. Gene editing could one day be used to treat obesity, say scientists at Harvard University. In August 2020, the researcher revealed a new method for combating weight gain in mice, transforming unhealthy white fat cells into energy-bursting brown fat cells using CRISPR gene edit. Stodgy white fat cells are full of unhealthy lipids that builds up inside the body. An excess of white fat can lead to diabetes, but brown fat cells are much healthier. They break down some fats to create energy and store the rest in a smaller place. The Harvard team was able to help the mice lose weight by altering their DNA. The scientists genetically altered the white fat, giving it the characteristics of healthy brown fat. The experiment focused on UCP1, a protein found in a brown rat that turns chemical energy into heat. Over the 12-week study, the mice with white fat cells piled on the pounds, whereas the gene-edited mice found it more difficult. There's even a suggestion that the gene editing process helped the mice starve off diabetes. Scientists predict that eventually this method could be developed into a treating of obesity, although human trials are still a long way off. Curing hearing loss through gene therapy. In 2019, researchers from Harvard Medical School and Boston Children's Hospital announced a novel treatment for hearing loss in mice that could one day be used on humans. Beethoven mice suffered from a genetic mutation that also affects humans, causing progressive hearing loss and eventual deafness. The name Beethoven mice is a reference to the German composer who began to lose his hearing when he was in his 20s. The hearing loss that the mice experience is caused by minor alterations in their DNA. Using sophisticated biological technology, scientists can pinpoint the defective gene without harming any of the remaining healthy genes. This means they can cure the Beethoven mice of their deafness without causing any unwanted side effects. The scientists do warn people not to get their hopes up too quickly. There are still years of research to be done before this therapy can be tried on humans. We believe our work opens the door toward a hyper-targeted way to treat an array of genetic disorders that arise from one defective copy of a gene, explained Harvard's Jeffrey Holt. This truly is precision medicine. That brings us to the end of the video. I hope you enjoyed the video. Thank you for watching, and we'll see you in the next video.